In Unit 6, we will discuss the ABO and H blood group systems. A blood group system is composed of antigens produced by alleles at a single genetic locus or at loci so closely linked that genetic crossing over rarely occurs. A blood group system is composed of antigens produced by alleles at a single genetic locus or at loci so closely linked that genetic crossing over rarely occurs. There are now 41 formally registered blood group systems. Your textbook has a few less, but that's because since publication, additional blood group systems have been discovered. Blood group antigens are molecules located on the red cell surface, or they may be an integral part of the cell membrane by crossing through the bilipid layer. And in some blood group systems, if these antigens are lacking, this could cause deformation of the red cell. They're classified as proteins and carbohydrates, and remember what makes a good antigen? Carbohydrates and proteins, and these are linked to either lipid or proteins. After exposure to blood group antigens, this may stimulate the production of a corresponding antibody. During a blood transfusion, an individual is exposed to many blood group antigens they do not possess, and this may result in the production of alloantibodies. Blood antigen nomenclature, or how we name these different antigens. Due to inconsistent use for terminology used for red cell antigens, the International Society of Blood Transfusion, ISBT, created a group to standardize blood group systems and names. The working party is called the Red Cell Immunogenetics and Blood Group Terminology and is charged not with creating replacement terminology, but rather to add terminology easily used with computer software. ABO has an ISBT number 001 and it has 4 antigens. H is ISBT 018 and has 1 antigen. The textbook uses both commonly used names and includes ISBT symbols and numbers. You are not responsible for memorizing the ISBT nomenclature, but I want you to be aware of it. The history of blood transfusion medicine is fascinating, and if you need a break from your studies, I encourage you to visit the links in the reference section of the course, which provides an overview of the history of blood transfusions. All types of sources, animal blood, milk, saline, human blood, and others, were used for transfusion. Why would they want to use milk? I don't know. Human-to-human transfusions were successful, but there were many deaths as well, which caused blood transfusions to be banned worldwide until the mid-19th century. The ABO blood group system was discovered in 1900 by Karl Landsteiner, and this was a game changer and marked the beginning of modern blood banking and transfusion medicine. It remains the most important blood group system in transfusion medicine to this day. Well, how did he do this? What he did was he had all his colleagues' blood drawn, and he separated the serum from the cells, and he mixed the serum of one with all of the cells from the other. And this was done for every person in the group, and they were able to discover group A, B, and O. Now, they theorized there was a 4th blood group, and his former students who became associates, von de Castello and Sturley, discovered the AB group in 1902. Landsteiner's rule and Landsteiner's law are terms that are used interchangeably. Landsteiner noted the presence of agglutinating reciprocal antibodies are consistently and predictably present in the sera of normal people whose red cells lack the corresponding ABO antigen. For example, an individual with A antigens will always have anti-B in their serum or plasma. If the corresponding antibody is not present, this would result in an ABO discrepancy. Landsteiner was awarded the Nobel Prize in Physiology or Medicine in 1930 for his work. This chart illustrates Landsteiner's rule, and you must memorize and know for the rest of your life what ABO antibodies will be found in the presence of each blood group. And so we have red cells on the top row, then the antibodies that are present, and the antigens. So if you are group A, you will have anti-B in your serum. If you are group B, you will have anti-A. If you're group AB, you will have none. And that is why for plasma donors, AB is a universal donor because they don't have anti-A and they don't have anti-B. And then group O people have both antibodies because they have neither antigen on their cell. And this is why group O is the universal donor for red cells, because they have no ABO or A and B antigens. It's important for you to have a basic knowledge of the red cell membrane structure. The surface of the red cell consists of a bilipid membrane in which large protein molecules are embedded. And they may not just be embedded, they may go all the way through. It's composed of phospholipids, which are both hydrophilic, water-loving heads, and hydrophobic, water-hating tails. The hydrophilic heads orient outwards into the fluid, both inside and outside of the red cell. The hydrophobic tails orient into the interior, away from the fluid. The top left picture actually shows you the red cell membrane, and if you look very closely, you can almost discern the little hydrophobic heads and tails. The line illustrations show you again how the membrane is made up and how the structures can protrude from and maybe even go all the way through the red cell membrane, which can then result in an important part of the structure of the red cell. The external surface of the red cell membrane is coated with a diverse array of glycoproteins, complex carbohydrates, and lipoproteins, imparting antigenic structure to the membrane. This is another picture that illustrates the same concept. Spend a few minutes looking at it. The inheritance of ABO antigens follows Mendelian genetics, which we've already talked about. And ABO frequency in the white population, approximately 45% is group O, 40% group A, 11% B, and 4% AB. You must memorize these statistics. This becomes very useful when you're actually working in the transfusion service. The frequencies differ in selected populations and ethnic groups. For example, group B is higher in Black and Asian populations. Here in Austin, we have a large Asian population, and whenever an Asian name came across, their frequency being group B was about the same frequency of white people being group A. So it was interesting. It's a little game I played with myself. We make our job fun to, just kind of wonder what blood group they would be. Many antigens vary in frequency in different races. This becomes important when looking for antigen-negative blood. So don't ever think there's anything involved with prejudice here. For example, there's a very, very high frequency antigen called U. The only ethnicity that you will find U negative blood is in the Black population. So you're not even going to bother testing other ethnic groups. The A gene codes for the production of a galactosamine transferase that affects the addition of N-acetylgalactosamine to the preformed H-bearing chains. So on the left, we see that L-fucose has been added to the precursor chain. This results in the H antigen. On the right, you see now that H has been formed, the A antigen or the N-acetylgalactosamine can be added, which results in the A antigen being produced. The B gene codes for production of galactosyl transferase. This affects the addition of D-galactose to the same H-bearing structure. So once again, I want you to know this is a building block approach. We have a precursor chain. Then we have the addition of H. No matter what blood group system you are, H is going to be present. If it's only H, you're group O. But H is the building block to which the transferase will cause the addition of the terminal sugar. So if it's D-galactose, this creates the B antigen. So here's a summary of the structures of the A, B, and H antigens. Again, I'm a visual person like we all are, and this to me pulls it all together. So we have the precursor substance, L-fucose is added, that's H. Then we have that basic structure with H, to which then we can have the A terminal sugar or the B terminal sugar be added. The H blood group system has two genes, H and little h. H is found on 99.9% of the population. Little h is found on less than 0.1% of the population. The inheritance of H leads to the production of the H antigen, which is the precursor molecule for A and B antigens. Again, remember, ABO antigens are built onto the cells due to the inheritance of a transferase. There is a lectin, Ulex europaeus, which has anti-H ability. And not all H is converted to A or B. It varies among the blood groups. And so we'll talk about this later, but you must memorize the amount of H from most to least. Group O people have the most because all they have is H. A1B people have the least because almost all of the H has been converted to either A or B antigens. The ones in the middle, A2, they are a subgroup system of group A, which we'll talk about, but the reason they're A2 is they have less of the H converted to group A, so they'll have more H, and so on. We're going to talk about H antibodies, or antibodies to the H antigen, and H-like antigens are found in nature. And anti-H is occasionally detected in A1 and A1B people, and rarely in group B individuals. And this will cause a false positive in the reverse reaction. The reverse reaction is where you've added unknown serum to your known cells. The reason for this is there's so little H on their cells that they may form an anti-H. It's relatively weak, which is the good news. So if you have a group A person, the B cells may react 1+. I've not really seen reactions stronger than that. It almost always reacts at room temperature. So what is the antibody class involved? IgM. Whenever you have room temperature reactions, it's an IgM class antibody. It's considered clinically insignificant, meaning even though it's there, the chances of it reacting with transfused cells and causing hemolysis are slim to none for a variety of reasons we'll get into later in the course. So let's talk about some issues that H antibodies may produce in ABO discrepancies. One thing I want you to do every single time is interpret your forward reactions and your reverse reactions separately, each and every time. This is why. Let's look at the first example on row 1. We have anti-A is positive, anti-B is negative. We have a group A person. In the reverse type, we have the A1 cells are positive, the B cells are positive. This is a group O. The forward and the reverse do not match. Let's look at the second example. We have positive with anti-A, positive with anti-B. We have a group AB person. Let's look at the reverse. We have a positive with the A1 cells, a positive with the B cells. They are reverse typing as a group O. And you say, Terry, it's only 1+. It should be stronger. Absolutely. Then we go to that next level of look at the strengths of the reactions, that they're very weak. And that will set us on paths of, maybe I have a weak anti-H, and I will do additional testing to prove that. So again, always interpret the forward and the reverse separately. And if they don't match, it's called an ABO discrepancy. And in anti-H antibodies, The reactions in the reverse type are usually very, very weak, but you cannot transfuse this individual until you resolve the ABO discrepancy. So we've talked about anti-big H antibodies. In contrast, persons of the rare O little h, or they have a double dose of the little h antigen, that's their phenotype, they have no A antigens, B, or H antigens on their red cells. And because of this, they form a very potent, clinically significant anti-H, which reacts well over a wide thermal range, room temperature through 37, and with all red cells except those of other O little h people. And remember, the frequency of the big H antigen is greater than 99.9%. So that means good luck finding a unit of blood. So let's look and see what their reactions would look like. In the forward type, negative with anti-A, negative with anti-B, they're group O, right? No problem. Reverse type, positive with the A1 cell, yep. Positive with group B cells, yep. What's odd is they're 4+ with an O cell. If I am group O, I should not react with my own blood group, but they are. This blood group is known as Bombay, and it's written O subscript little h. If L-fucose is not added to the precursor chain, then H, A, and B cannot be added. Remember, blood groups are built onto the red cell by means of transferases which attach certain sugars. So without L-fucose, then A and B also cannot be added. The individuals may inherit A or B genes, but without H, the immunodominant sugars cannot be added. And this can cause a really big deal in paternity testing when you have a group O mom and a group O dad, but you have an A baby. And this could be that they may be this very rare genotype of Bombay. You must be homozygous for little h, which is an amorph with no detectable product. It's similar to little d. If H is present, H will be expressed, no problem. But if you're homozygous for little h, big problem. A Bombay individual has the genotype little h, but will type as a group O. Because there's no A or B antigens on the cell in the forward type, you get group O. In the reverse type, because they don't have A or B antigens, they'll be potent anti-A and anti-B. So yay, they're group O. But it's discovered due to incompatibility with all blood groups, including group O, because you should not react with antigens that you possess on your red cells. So this would indicate the absence of H. This can only be determined by family studies or testing with lectins. So the Bombay genotype occurs when two little h genes are inherited at the H locus. Again, you will possess normal A or B genes if they were inherited, but you're unable to express them because you must have H on the red cell membrane to express A or B antigens. They can transmit A or B genes to their offspring. The term Bombay was used since it was first discovered in Bombay, India. And we found out that we have a Bombay living in South Texas, and our prayer was, please don't ever move to Austin. I really want you to understand the genetics of the Bombay genotype. So these individuals, remember they're little h, they can genetically be a group A or group B, but because they can't build H, there's nothing for the transferases to attach the terminal sugars to. So let's look at this pedigree. We have a group O and a group B, and they have two group O's and one B children. And this is possible because the individual is a BO, which means they can transmit a group O to the mix. Let's look at the next generation. We have a group O and a group A who produce an AB child and a group O child. This is not possible. The group O, maybe. The AB, absolutely not. There's no way a Group O person can give birth to an AB child unless they're a Bombay. And so this Group O person is a Bombay. How do I know that? Because they were able to transmit the Group B gene. The mom gave a normal H, and the dad was able to. Let's apply our knowledge of genetics to the Bombay genotype. So a Bombay individual, again, they can pass A and or B antigens to their offspring with normal H, which may lead to great confusion. So in the first example, we have a Group O dad, a Group B mom, and the offspring are two Group O's and a Group B, which that makes sense because mom can be a BO. In the next generation, we have the son who's Group O, who marries a Group A lady, and they have two children. One is an AB and one is an O. The O is absolutely a possibility, but the AB is absolutely not, because a Group O person cannot have an A or B child, much less AB, unless they are a Bombay. And again, they have inherited a B antigen from their mom, but because they have no H, they can't express it, but they can transmit it. So if you have time, the Blood Bank guy is wonderful. He has so many podcasts. He has so much material. I've referred him to my students for years and years and years because he brings things down to a very basic level. So whenever you're struggling with a topic, go to the Blood Bank guy. So again, OH denotes the phenotype of the Bombay. The red cells are not agglutinated by anti-A, B, or A, B. The serum or plasma agglutinates A and B cells. It's not recognized until the serum is tested against Group O cells and causes strong agglutination. These individuals have 4 ABO antibodies in their plasma. They have anti-A, anti-B, anti-A, B, which reacts with both A and or B cells, and anti-H. They can only be transfused with Bombay blood, which statistically only less than 0.01% of the population lacks the big H antigen. Confirmatory testing to determine whether or not a person's Bombay is by using the anti-H lectin, Ulex europaeus. You must memorize that. We'll talk about lectins in a moment. Because this is directed against the H antigen, if this person is a true Bombay, it will be negative. Agglutination of A, B, AB, and O cells by the patient's serum or plasma, and no agglutination when the individual's serum or plasma is tested against Bombay cells. This is the end of part 1 of the lecture.